There seems to be this pattern of if women are very low in fish intake or blood levels of DHA or omega-3s, then the intervention helps. But if they aren't low, they come in with good levels, they come in eating fish, then the intervention doesn't actually, it's not different than the placebo. And to me, that's that blood level thing.
Welcome to the Proof Podcast, a space for science-based conversation exploring the health and longevity benefits that come with mastering nutrition, physical exercise, mindfulness, recovery, sleep, and alignment. Facts, nuance, and trustworthy recommendations, minus the hyperbole. Hi, Dr. Harris. Thank you so much for joining us today.
Thank you. I'm so happy to be here.
Yes, I had your dad on recently, and that episode hasn't gone up yet, but it will before this one goes up. It was a great deep dive, as I'm sure you can imagine, into omega-3s and in particular how they affect cardiovascular and neurological health in adults. Today, I thought we could focus a little more on the maternal and infant periods and the importance of omega-3s in these life stages, which I know is an area of interest for you. Perhaps the best kind of on-ramp here is how you personally wound up studying nutrition, and I'm assuming as a kid and a teenager, your dad probably shared a lot of his work and thoughts about nutrition at the dinner table.
Yeah, I never really expected to do this, I think. I think it was kind of a surprise to me. But the biggest thing I remember about my dad's job growing up is that we were able to travel a lot with him, because in academia, everything's really international. And in the omega-3 world, there's a whole lot of connection to Norway and Scandinavia. And so when I was a kid, we actually lived in Norway for 6 months while he studied with another scientist there. And that was an amazing experience, but it also was like, this is kind of an interesting lifestyle. That's very cool. I liked it. And then as I went through school, undergrad, I was at a liberal arts school and I studied biology. And I really liked biology, but I did not want to be a medical doctor. I didn't want to have life and death decisions on my hands, but I thought metabolism is interesting. And my dad basically was like, well, metabolism is really close to nutrition. Nutritional biochem is metabolism. So I wasn't really sure because I took maybe one nutrition class in undergrad. They didn't have anything upper level. It was a small school. And so all I knew really was kind of calories in, calories out type of nutrition. And so I did apply to grad schools, and I ended up going to the Penn State, which was far away from where I was in South Dakota. I went to Pennsylvania. It's 12 hours, or it's 20 hours away driving. And I didn't know anybody out there. And I just went and I was like, we'll just see. If I really like this, I'll know. And if I don't, I'll just be done. And it turned out I really, really liked it. And I had a really great mentor, Penny Chris Etherton. She was very kind and had lots of interesting projects. And I really thought I just loved my classes. And so it was I kind of backed into it a little bit and just explored and figured out well, this is really interesting. While I was at Penn State, I did my PhD in Nutritional Sciences running human clinical studies that were feeding studies, which was really interesting. And it wasn't in omega-3s at all. And then I also got my registered dietitian credentials. So I got the really clinical side of things, which I really like. And then after I did an internship, I did a postdoc in behavioral health, which was kind of going more towards the public health. How do we just make small changes on a big scale? Totally going away from biochemistry. And then I realized after about a year of that, I was like, I actually don't like this as much. I feel out of my depth. It is so many variables. It's way beyond nutrition. So I decided to come back into nutrition. And I had worked for my dad's lab, which is called OmegaQuant. And they've been doing fatty acid testing for years. And I came back to it and I was like, I'm interested to try working here again, and see if I like this. And when I came back on board at OmegaQuant, they had just validated a breast milk fatty acid test. And they're like, well, we're able to measure fatty acids in milk. So we're really interested in looking at maternal health. And I was like, okay, I've never done maternity or maternal health stuff. I hadn't had kids yet. But I just kind of started to dive in, and it was really interesting. There's heart disease is where a lot of our main data is, but I think the maternal piece is almost as strong as the cardiovascular piece. So from the milk DHA levels, as I was looking at that and trying to determine kind of cut points, I kept saying that pregnancy is really where a lot of the big effects are. And so the next thing we did was, well, let's look at what omega-3 levels in pregnancy mean. What kind of outcomes are there? What's the cut point? And so that's really how I got there. Kind of a long-winded answer, but I always kind of just do the next right thing. And I didn't ever really have a full plan.
Yeah, I think you kind of reeled that off quite nonchalantly, but that's a seriously comprehensive education. Yeah. I'm curious, and some people listening may not be familiar with Penny Chris Etherton, but she is a real giant in the field of nutrition science. Mm-hmm. It doesn't get much better than sort of cutting your teeth under her supervision. Can you share what that was like?
Yes. So for her she is a giant in our field, but she is like a small, kind woman. She is has a small demeanor, but she's so kind, and she was able to do so much work and get so many students through her lab and through the graduate school without kind of having any academia ego, which I think is really, really good. And it was just a really healthy environment to learn and make mistakes, and she's very human with everyone. And so, yeah, she had a lab where we were doing controlled feeding studies, which is a really hard, high-level way to do nutrition research, but we were able to really see how to do that high level of nutrition research on all kinds of different areas of human health. But she really was deep into the cholesterol story and fats and fatty acids in the beginning of her career and throughout, but she's definitely branched out and done lots of other things too. Because my study was really focused on kind of whole grains, but other people were doing a lot of nut studies and different. There were some omega-3 studies, there were some cholesterol studies, but mostly she really did her work well. She was very thorough in reviewing our work and giving really good feedback, and just made it easy to want to do the next thing for her. It was like, I'm excited to do this study. And so much of it is you don't know what you're doing. So much of it is that human element of her just being kind enough to let you make mistakes or let you learn along the way, let you ask questions, and then having that expertise to really guide you in the right way while still letting you make some of your own decisions. But it's very fun to go to conferences with her because they know everybody, and everybody just loves seeing her. So
you did your PhD in looking at whole grains, is that right?
Yeah, whole grains.
And I know this might be kind of a slight change in gears and going away from today's topic, but can you tell us a little bit of kind of what you were interested in looking at in your PhD and what you found?
Mm-hmm. Yeah, so we were really looking at metabolic syndrome, and my study was a follow-up study to a previous one where they're basically taking like the same diet and switching a refined grain product for a whole grain product. And so that was it's fairly simple, but there's still so many variables changing that it's really gets tricky. But we were mostly focused on those metabolic syndrome characteristics of blood glucose and blood pressure and waist circumference. And so we started to, I would say what we found was that there were some differences. There were some improvements in like overall metabolic syndrome scores. on a whole grain versus a refined grain diet, but it was not super dramatic. And I would say part of that is the whole grains we were studying were still at the same level of processing. So it wasn't like we were taking a whole, they weren't having like farro versus pasta. It was like whole grain pasta versus pasta. And so that element always to me was like, that's that next step of kind of a less processed whole grain, but it was still interesting to do the processed ones because that's what's kind of realistic. And so it was, we did resting metabolic rate. We did MRIs to look at visceral fat. We did all kinds of blood work. And we found some interesting findings, but overall it was a pretty small difference, I think, between the groups. And which I think is probably kind of to be expected because all of the groups came onto a controlled feeding study from a free living diet without a run-in period. And so there's automatically a difference when you have someone feeding you and how you act in your behavior. And we were trying to do a weight maintenance for the first part of the study and then weight loss at the last part. But it was very hard to keep people their weight up once you're controlling their diet. They're still getting like, some people were taking like bags of lettuce home and trying to eat it. They couldn't eat all the food we were trying to give them to maintain weight. And so that's something that was a big learning experience for me, is just like that, just the controlled feeding element is such a big variable. And so, yeah, so we found whole grains did some interesting things. A little bit of improvement in metabolic syndrome outcomes, but not wildly dramatically different.
So it kind of sounds like weight loss was driving a lot of the changes, and that was independent of the type of grain.
Yeah. And it's really, I mean, that's the key. It's hard to control that. And once you bump up all that fiber, it's really hard to get people to eat enough to be able to maintain that weight.
But your point about different types of whole grains, that's also interesting because we kind of fall into this trap of just having umbrella terms, but there is likely a real difference between different types of whole grains, as you say, and the relative degree of processing between them. It's super interesting. Yeah. You alluded earlier to the kind of benefit of having the benefits of having your dietetics training. And now as a scientist, I'm curious as to how you feel that benefits you in terms of nutrition science and thinking about experiments, conducting experiments, and sort of coming to conclusions as well.
Oh, that's a good question. It's a big question. To me, I think it just reinforces the humans that are in science, both running the scientific studies and as participants, and the human behaviors that people that really do kind of affect scientific outcomes, and how study design is really trying to take away as many of those human elements as possible to really see, like, is the intervention doing something? And so as an RD, your training is working, it's really focused on the individual. It's focused on their behavior, and their entire, it's just so much bigger than food when it comes to nutrition. It's everything in their lifestyle that allows them, to have the money to get the type of food they want, the skills to cook it. There's a lot of different variables besides the nutrition. So getting that dietetic training, really exposed me to all different kinds of ways you can practice nutrition. But I think the bigger piece that I bring into being in research is that individual variability is real in studies, biologically, as well as the compliance issue. And so people will tell you what you want to hear because they're trying to be good patients or participants, but they don't necessarily want to say that they've been not doing something according to plan. And then you can't, as a dietitian, if you don't know that, you can't help them, which is a very human thing, I think, for us not to admit we're not doing something right when we know we should do better. I do that. I'm behavior and knowledge are different things. And then the same in research is that's why you have so many different tests you do, is to make sure you're trying to, decrease the amount of differences between each participant in your study. So a controlled feeding study, you're giving them all your food every day for 3 months. That's a huge amount of effort to just try and control that one piece. And still you don't know what they're actually eating. You still don't. They're not. There's another level of research, nutrition research, where they're stuck in a room for a week or months or something like that. Like, it's a whole nother level. So I think the humanity, it lets me come at research findings and studies with a grain of salt. I'm never like, this is going to save everybody, because it's just, we're way more complicated than that. And so I think everything taken with a grain of salt, is what I've learned from dietetics and working with humans.
Yeah. And I guess those very, very controlled environments, in kind of metabolic ward environments, as you say, kind of locking people away, are very good for controlling as many variables as possible, but then are perhaps not as generalizable in terms of what happens in the real world in a sort of free living scenario.
And every different kind of study design has a purpose, 100%. Like, we might kind of look down on dietary intake studies where you're asking people to recall what they've eaten over the last 6 months or the last 3 days. But there's a lot of error in there, but it's also like, you also can reach so many more people. And that's a really big advantage. It's like, the higher the quality of your study, as far as compliance and control goes, the smaller amount of people you have. And the less realistic it is and less broad it is. And then the more broad it is, usually you lose that level of quality control because it's almost impossible. So that kind of leads into omega-3 testing, and how one of my favorite things it does is it shows like an individual biomarker response to a nutrient, which is not all that common to have a really nice nutritional biomarker.
Yeah. I want to talk about why that's important and how that can be potentially helpful in various studies. But let's take one step back and kind of assume that someone's jumping into this conversation perhaps with very little background knowledge of omega-3s. Perhaps they haven't listened to the episode that I did with your dad, Bill. What are omega-3s? What are the kind of different types?
Yes. Omega-3 fatty acids are polyunsaturated fatty acids. They're a type of fat. They have... fats have a lot of carbons, and they also can have different amounts of what we call double bonds. And that, if a fat doesn't have any double bonds, it's really straight. If it has one double bond, it turns at that, and it starts to get, we call it usually they call it kinky. It just can be like lots of different shapes. But it always feels weird. And so polyunsaturated fats are very long, and they are lots of different shapes because they have a lot of different double bonds. So unsaturated means double bonds, poly means many. So monounsaturated only has one double bond, and olive oil is like your main example of that. And the other thing, it's structure and function. So in biology, the structure of something really tells you about the function of it. And so a saturate versus a monounsaturated versus a polyunsaturated, they have different structures, so they have different functions in the body. And one of the most obvious ways you can tell the difference between these fats and that they are physically different is that room temperature state. So butter having lots of saturated fat is solid at room temperature, and anything that has unsaturation is liquid at room temperature. So that's kind of that fluidity idea is actually really important in the body too. So omega-3s are a type of polyunsaturated fat. Their first double bond is from the 3 carbons from the omega end. That's the technical reason it's called omega-3 or omega-6. And those two types of fatty acids are kind of essential. Like, our body can't put a double bond on that 3 position or the 6 position. And so that makes something essential because your body needs that for a lot of different purposes. And then it can make lots of different omega-3s and omega-6 fatty acids based on getting the parent omega-3 or omega-6 through the diet. So your body's still metabolizing and using these fats, but at the core, we do need to get it from the diet. So we go zoom in even more. The omega-3s, we have alpha-linolenic acid, which is the omega-3, the parent omega-3. It's found in plants. It's found in walnuts and flaxseed. And that is one kind of omega-3. And then the other main ones are EPA and DHA. Those are the ones found in fish. We call them marine derived. They're really unique to fish, which is fascinating because there are not a lot of nutrients that are so specific to a type of food group. And those 2 fatty acids have a lot of carbons and a lot of double bonds, and they have been shown to be really important for health, for how our cells function. And so we can make EPA and DHA from ALA, but it's a pretty slow process. And there's a lot of different things that can affect it. But we do know that when we eat fish or we eat EPA and DHA in a fish oil, the EPA and DHA in our body goes up. And it's a pretty direct relationship. And that's pretty rare. A lot of times the body takes whatever you give it and it makes whatever it needs. And for EPA and DHA, it seems like that connection, it's pretty direct between diet and what's in our blood. So, that's it's another long answer to what are omega-3s.
No, that was great. And we'll save omega-6s potentially for the sort of end of this conversation if there's time. You mentioned there that omega-3s, there's research that has looked at the health benefits of omega-3s, ALA, DHA, and EPA. If we zoom in now on pregnancy, that sort of maternal period, what do we understand with regards to omega-3s and their importance during pregnancy? I'd love to kind of go through what we know and how that's been established.
Yeah, it's a big question. I'd say, I think 30, maybe even 40 years ago, this question around Dr. Olsen, in, I think it was Copenhagen, he started to look at the Faroe Islands, which is like a tiny island in the middle of the North Atlantic Sea or ocean. And, so they didn't have much food besides fish and fatty fish. And EPA and DHA are really present in cold water fish. So they had very, very high levels of that. And then they were comparing that population to a typical Danish population and the pregnancies. And they found that the women on the island had much larger babies than the women who were in Denmark. And those women, they started to do, and that just kind of started the process. So they started to do research. They found that their gestation was elongated. So they were having longer pregnancies on average than the Danish women were. And that would make sense. A bigger baby, the longer the baby has to grow in the womb, the bigger it should be. And so then they started to ask about fish intake, and they saw that women who ate 3 or more times of servings of fish a week had longer gestations than women who ate less than 1 serving or 0 servings a week. And so they keep like going down this path, and this group, I don't know how to say it's Sigford Olsen. They've published so much in this area. So they really started this focus on kind of gestational length and just which is a really typical thing to look at and measure in pregnancy. It's one of the most basic things you look at. And then there was also a turn where DHA specifically is very rich in the brain and in eyes. It's much, much richer than the rest of our body. And so they started to see that with premature babies coming out, they would have eye issues or brain development issues. And especially if they came out and were put on a formula that didn't have any DHA or arachidonic acid, which is an omega-6, both really important. So they started to do research on things like visual acuity and giving pregnant women doses of fish oil, so concentrated EPA and DHA from fish. And a lot of these studies happened in the Nordic countries as well. So they already had a pretty high level of intake. And then they really did some very high dosing studies, like 6 grams a day during pregnancy. And they did it in some really high risk pregnancies too. And they found this pattern across the board, even in these high risk pregnancies, that women who had low fish intake had a benefit from taking the fish oil, so a longer gestation or other types of pregnancy benefits. But women who had high fish intake didn't see a difference if they were on the fish oil or the placebo. And there were some other groups, Bob Gibson and Maria McCrides in Australia. They did some very big studies on postpartum depression because they started to see some signals that women with very low fish intake or very low omega-3 status would have more issues with postpartum depression. And they did not see, when they did their big study on that, they didn't find an effect on postpartum depression, but they saw that there was a reduction in premature birth, and early premature birth specifically, which is earlier than 34 weeks gestation. And the earlier you give birth, the more complications. So a typical premature birth is 37 weeks. I mean, these are the cut points that a lot of this research uses. There's no official cut point from what I can tell. It can go around bump around a little bit. 40 weeks is term. I think we can all agree on that. And so they found, they were looking at postpartum depression, didn't really see a big effect, but they found this effect on premature birth rates. And then at the same time, a group in Kansas City, Susan Carlson's group, they were also doing a study, trying to think of what their primary outcome was, but they were doing a DHA outcome study, which I think had, they wanted to look long-term at like toddler, like the baby's development, cognition, and all these different outcomes. And those outcomes are more mixed, but they found in that study a reduction in early preterm birth and a reduction in NICU days for babies who were in women who had the fish oil supplement versus women who were on the placebo. So you kind of have all these different groups coming together. They're all kind of interested in different things. But then it starts to come together that a really clear outcome is being able to elongate gestation, which means you have less preterm birth. And preterm birth is a huge issue. It has so many effects. The earlier baby comes out, the more it needs just to get on the like normal track. It's incredible what our NICUs can do to catch babies up who have to be born that early. But it's a huge effort. It's a huge amount of money, and it's a huge amount of stress. So being able to not have preterm birth is desirable. So all of these started to come together. And in 2019, I think, the group in Australia, the McCrides group, they published a Cochrane report, which is a really high-level meta-analysis that has a lot of rules to make sure you're not being biased. And they found really strong effects on omega-3 arms or fish intake arms, reducing risk for preterm birth, especially early preterm birth. And then that kind of, for me, that was the paper that was like, okay, we need to make a DHA target for pregnancy. But all of these coming together, it's basically like the omega-3s are doing something to decrease, to elongate birth, to decrease early contractions, something in that area. We can talk about potential mechanisms, but even preterm birth, the mechanism of that is like a little, it's not clear. But there's all these other side studies on visual acuity, on toddler cognition, on infant cognition, on heart rate variability, on, all these other things that make sense. That there's a real it's a brain or an eye connection typically, or like a immune function or autonomic function focus. But there's there is another, besides the elongation of gestation, the other main finding that you see across several studies is that if they look at baseline either blood levels of omega-3s or fish intake, there seems to be this pattern of if women are very low in fish intake or blood levels of DHA or omega-3s, then the intervention helps. But if they aren't low, they come in with good levels, they come in eating fish, then the intervention doesn't actually, it's not different than the placebo. And to me, that's that blood level thing. And so that's kind of, there's so much more than that, but I'm trying, it's a huge field. It's just huge.
I've got a few questions so we can perhaps dig into that. And I want to make sure we emphasize what healthy blood levels look like versus levels that may be less than optimal. You mentioned DHA quite a bit there, and this sort of clear signal of benefit with regards to period of gestation and the weight of a baby. Is it just DHA? I do want to try and get into what are some of the proposed mechanisms, but often we hear both DHA and EPA coming up in these conversations about omega-3s. When we're talking about pregnancy, are we more interested and focused on DHA, or is there also some science looking at possible benefits of EPA as well?
I'd say it's probably more of the literature probably is using fish or fish oil, which always has EPA and DHA. But there have been studies using pure DHA. And I think a couple of reasons for that was they were able to get pure DHA manufactured much earlier than pure EPA. And that was primarily for infant formula, because that is a whole nother area where they experimented with infant formula for premature and term babies. And the other thing is, it's just, I think it's as simple as there's a ton of DHA in your brain and your eyes. And so there have been animal studies where they completely eliminated omega-3, specifically DHA, and over time there's just major dysfunction in brain and eye development. But their focus kind of became on DHA, especially in the studies that were focused on brain and eye outcomes. And then the reporting, if they reported blood levels, a lot of times they're only reporting DHA. And in the omega-3 index, which is a metric of EPA plus DHA in red blood cell membranes, the DHA portion is typically 85% of that. DHA seems to be much more structural in the body, and EPA maybe is used up faster. It might get metabolized a lot faster. And so there's just a lot more signals with DHA. And then the other thing is we have some specific dietary recommendations for DHA alone and not for EPA. And those are a lot of the reasons I started to look more specifically at DHA than the combination when I was trying to just see what the literature said. Partially, it's just what's been reported is just DHA a lot of the time. And the really big studies that have recently been done just use pure DHA as their intervention.
And what do you think some of those proposed or potential mechanisms may be where DHA is having an effect on gestation length?
Yeah, so these are hypotheses. So this is just what I've gathered. But so omega-3s and EPA and DHA have a lot of metabolites that are really important in the beginning of pregnancy or in the beginning of labor. So I think prostaglandin E, prostaglandin F2, and those are related both to DHA and EPA as well as arachidonic acid, which is an omega-6, also really important. But there is those molecules are incredibly important in the contraction, starting the contraction of the uterus. And the uterus contracting is labor and delivery. Like that's how the baby gets squeezed out of the body. So those signals starting to happen are related in some way to these fatty acids because they are made from these fatty acids. And just generally in anyone's body, when we have these fatty acids in our cell membranes, in our tissues, the body takes them and metabolizes them into an amazing array of different kinds of metabolites, which are really potent. Many of them on the omega-3 side tend to be anti-inflammatory or pro-resolving. So after an inflammatory process happens, the body has to heal itself essentially from that process. And many of the omega-6s, their metabolites tend to be in that first part of the process that's more pro-inflammatory. And the omega-3s tend to have the ones that come and clean up afterwards. And so I think of preterm birth kind of as an inflammatory event. Like there's been some research that shows like a virus or having some kind of virus can kick off early labor. All of the risk factors for early preterm birth all point to higher stress. And stress adds more inflammation into your body. But they're very vague. They're like being overweight, being underweight, being older, being too young. And then it's just generally having like more stressful lifestyles. So if stress or inflammation or a virus, which obviously kicks up inflammation, those two things kind of have an association with preterm birth. That to me is kind of a little bit more of an inflammatory environment than you want. And then many people are very low in omega-3s in their body, in their status. And so I don't think it's just omega-3s happening, but I think it's one of the pieces that's when those are really depleted, then your body just starts to not be able to fend off some of these different challenges. And then they're intimately involved. Their metabolites are so intimately involved in the actual contraction of the uterus, which is preterm, which is birth. I think it's in that arena where it's more about inflammation than necessarily structure or fluidity of the ability of the uterus to stretch, because we don't see that twins, their gestation is affected by omega-3 status. That stretch factor, which is also a trigger for birth, is not really related to the omega-3 status. So, so in my mind, it has a lot more to do with those prostaglandins and eicosanoids becoming metabolites of these fatty acids. Something gets off, and I think inflammation is some kind of trigger.
Yeah, that's fascinating about twins. You mentioned omega-3 status there a few times. Can we kind of quantify that? When you say a healthy status or an optimal status, what do you mean? And how could someone kind of check what their status is?
Yeah, so this is a big deal. It's not standardized. So this is something that I hope that the recent research and push in this field can really help omega-3 testing get standardized. So omega-3 status just means omega-3 levels in the body, but there are a bunch of different ways you could measure that. And you're not going to get the same answer across the board. So I'll just take at OmegaQuant, we measure the omega-3 index. And the omega-3 index was invented by my dad, Bill Harris, and Clemens von Schacke, who's a cardiologist in Germany. And it was looking at a red blood cell membrane and seeing how much EPA and DHA is in that membrane. Because membranes, like cell membranes, are made up of fatty acids. And there's lots of different kinds of fatty acids in there. We measure 24 different kinds of fatty acids. And we'll take the EPA and DHA over the total and get a percent. So they found for heart disease that having a 4% omega-3 index was a much higher risk than having an 8% omega-3 index. For really sudden cardiac death was the main outcome originally. So 8% of your fatty acids in your red blood cells as EPA and DHA seems to be a really healthy target. But if we then that 8% can't be applied to a plasma phospholipid level or a total plasma level of omega-3s. And a lot of different labs will measure plasma levels. And so their cut points are going to be coming from something else. And it's really confusing, and it's a problem. If we don't get this standardized, we won't be able to use omega-3 testing broadly to actually help any of these conditions. And so for the pregnancy status, DHA levels, when we looked at it, we saw that women who had a DHA level, just DHA, of 5% in a red blood cell membrane were really at quite low risk of early preterm birth. So we tried to look at the literature to see what are the outcomes in pregnancy for early preterm birth as our outcome. And 5% kept showing up as the point where after that, if you get higher and higher levels, that preterm birth risk doesn't increase. It kind of, the risk line kind of flattens out. Doesn't mean that being higher than 5% DHA doesn't have other benefits. If you're at 5% DHA, you probably have at least an omega-3 index of 6. But having an omega-3 index of 8 is probably just even better for a lot of pregnant women. But we didn't have evidence that getting higher and higher was improving preterm birth outcomes. So we were just trying to keep focused on that outcome. But below 5%, once you start going to 4% and 3%, the risk for early preterm birth was just very steep, very dramatic. And so it was really interesting because we proposed this 5% target for DHA. And a couple of things really kind of came together around this number. One was that 200 or 300 milligrams of DHA a day is really the recommended dose for pregnancy, to have at least 200 extra of DHA, 300 including diet, at the very base level. And we found that amount of intake, which is equivalent to about 2 servings of fish a week, which is the current recommendation for pregnant women in fish intake. That level is, it's about equivalent to 5%. So if you're eating fish twice a week, it's possible for you to be at 5%. Fish twice a week will not get you to an 8% omega-3 index. 8% omega-3 index is like a pescatarian diet or a 1 to 2 grams a day of fish oil. 5% DHA is 2 or 3 servings of fish a week or 300 milligrams of DHA a day. So that's a pretty big difference. And as a dietitian, I think that's really important, because if we came out and said every pregnant person needs to be on a gram of fish oil, which I think would be great, but that's a huge burden. That's a lot of fish oil. It's very expensive, and there's just not evidence for it needing to be that high. So that 5% target had some other rationale around it that had a dietary connection. And then in a recent study by Susan Carlson's group in Kansas City, they actually had a study where they did a 200 milligram DHA versus 1,000 milligrams of DHA a day with an outcome for preterm birth. And they found an overall effect. But when they looked closer, they found that women who had a red blood cell DHA level of 4.8%, almost 5%, that was a really important level. So they found that women who came in above 4.8%, it didn't matter if they were in 200 or 1,000. Their preterm birth risk was the same. And it was lower than the other group. The group coming in with a lower level, if they got 1,000 milligrams per day, their risk was half of the women who had 200 a day. So coming in with a low level, you really do need that higher rescue dose of 1,000 milligrams a day. 200 is not going to do it if you're coming into pregnancy and you're already depleted. And so we found that 4.8% was so close to the 5 that we found, where it really shows it's just that cut point where you're either replete or you really need a higher dose. And that's where the testing during pregnancy could really be helpful, because you're not going to recommend 1,000 to everybody. And it won't be appropriate for everybody and their lifestyle, but 200, 300, everybody should get that recommendation. But it allows for some real personalized nutrition counseling, in my opinion.
Sure. That's really, really reassuring, and some good targets in there. So 5% if you're wanting to kind of reduce risk of preterm, 8% to reduce risk of cardiovascular disease and kind of health benefits aside from pregnancy. Can we go back? You made a point earlier. I just want to reiterate this. You sort of established this point that these percentages we're talking about, these are specific to the omega-3 index, because I do think a lot of people would be getting standard labs with different kind of omega-3 tests. Can you possibly explain why we can't use these targets that you're talking about for more traditional omega-3 tests?
Yes, absolutely. And that comes from this red blood cells are a really neat thing to have a biomarker in. So the omega-3 index or omega-3 or red blood cell DHA, either of those are similar to HbA1c. So they are about a 4-month, red blood cells live for about 4 months in most people. And that level of omega-3s or of blood sugar that's been in your body for the last 4 months really shows up in the red blood cell. It's a long-term status marker. Plasma is the other part of your blood, and plasma has a different function. When it is high in omega-3s, it's usually because you've eaten them a few hours ago and it's just gone through your GI tract and you've absorbed it. And now it's high in the plasma because it's getting carried around the body, or it's going wherever it needs to go. But it'll go down after like a peak absorption is like 4 hours, and then it'll keep decreasing over the day. So if you're measuring in plasma, it can work if you are maybe consistently taking a fish oil. Then you may consistently have this higher and higher level of plasma fatty acids. What the actual targets are in plasma would be completely different than a red blood cell. They might be using a concentration, likely using a concentration if it's total plasma. They might be there's also plasma phospholipids. And so phospholipids, it's really like the red blood cell. The membrane of the red blood cell is phospholipid. That's how the fats are structured. And it makes the outside of components in the blood and in plasma. So it'd be like the walls of an LDL particle or triglyceride particle. So that plasma phospholipid level is actually used in a lot of research, because just fun fact about research is usually when you're doing a study, you just save all the plasma when you're taking blood from people and you throw away the red blood cells. It's very typical because it's just red blood cells. There's nothing interesting in that. All the proteins and all the fat levels are all in the plasma. Everything's in the plasma. The red blood cells, I think it's just out of a complete, they're just not used as much. So we don't have as many studies saving them. So we don't have as much data. So a lot of studies will use a plasma phospholipid because it has the same kind of longer term status marker as a red blood cell, because they're both phospholipid measures. But it still won't be that same scale of 4 and 8%. It'll be a different scale. It'd be like 3% is a good number. And so not only that, but like every lab uses slightly different methods. We did do a kind of a round robin study with other research labs in the fatty acid world, and did a comparison. And most of I think all the labs in that study came back with very similar omega-3 levels when they measured the same sample. But those are research labs. They are not available to a normal clinician. And so we were really I think we were the only clinical lab involved in that. And so we don't match. The labs aren't matching across the board. We're not measuring the same stuff. We don't even measure the same fatty acids. There's more than 24 different kinds of fatty acids in red blood cells. You can go you can, I mean, especially with the technology today, you can find all kinds of really specific fatty acids. So we need to, I think, hopefully the research community can come together and say, at least in research, we're going to measure these fatty acids in this blood, in red blood cells, or in plasma phospholipid, and we're going to report it this way, because people also report it differently. They'll either do an omega-3 index is EPA and DHA. Total omega-3s would be ALA, APA, DHA, DPA. Some people do EPA, DPA, DHA. There's no rhyme or reason. So, and they all are kind of singing the same song, but you don't have actual levels that you understand how to get to. So in almost every other outcome we look at, like HbA1c, there's a target. 5.7% is below that is considered normal. But we don't have that in the omega-3 space, because it's a really complicated method. It's hard. It's simple, but it's hard to do. It's not in plasma, requires different equipment than most labs have. So it's a specialized analysis, but it's been around for 30 or 40 years. It's the same type of analysis. But it's got a lot of issues. And so at OmegaQuant, our goal is to be as clear as possible with what our numbers mean. We do a lot of research. We work with a lot of research partners, academia, and pharma, and we work with healthcare providers so that they can get numbers that actually do compare to these big research studies, instead of just, you can get any omega-3 marker. If you do a pre-post and you take fish oil, you'll see lower and higher. You just won't have that connection to what am I actually shooting for? How high do my levels need to be? What makes sense? So there's a lot of work to be done to get this universally standardized, but for all of those reasons, there's a big difference between omega-3 status and across the board. So you really have to know what you're looking for.
Sure. So the main point being for the listeners here is that these percentages we're talking about are from the omega-3 index, and that is giving you an idea of your average or typical intake is sort of less susceptible to volatility in your intake, which may show up on one of the other tests if you're not consistent with your omega-3 consumption. I have a couple more questions here with regards to sort of optimal intake. You mentioned there 200 to 300 milligrams of DHA, or a couple pieces of fatty fish being that sort of recommendation to reduce risk of preterm infancy. I'm curious about kind of genetic variation in conversion of ALA to DHA and EPA. I've read a few different takes on this. Many people are talking about during pregnancy that conversion gets ramped up a little bit, and some people may convert more, some may convert less. Is it possible, do you feel, for some people without a direct source of DHA and EPA, if they're consuming adequate or optimal amounts of ALA, to get to that 5% omega-3 index level?
Gosh, that's a great question. As far as I know, the study in pregnant women has not been done to see, like, can ALA get you there DHA wise, but maybe it has. I'm just not thinking of it. So first, yes, estrogen increases the enzymatic pathway to get to EPA and even DHA, which is really interesting to me. To me, that is another like biological sign that these omega-3s are really important, because the body's ramping that up. And so when we say it ramps up that process, usually you're looking at 1% or less increase of DHA or EPA. Typically, what we can see when people take a lot of EPA or ALA, their EPA levels can increase, and even their DPA levels can increase. And DPA is in between EPA and DHA on the pathway. But it's usually stopped at DHA. DHA usually doesn't move. So we typically see the ALA could increase EPA, but not DHA. And so, but there's a lot of people experimenting with different parts of the diet. And I wouldn't say that it is an impossible thing for just for a total plant-based diet to reach 5% DHA. I just haven't seen it. And from what we've seen in the kinds of diets that are kind of typical here in the US, at least, and Australia and Europe have a lot of research in this too. Typically it does take DHA to see DHA move. There's going to be different levels of DHA, and that's at kind of baseline. So people's just normal diet, they're going to have a little bit of variety in DHA levels. Typically that is really impacted by fish intake, but there's other things that also impact it. We don't ever really see anything below 1 or 2% DHA red blood cell. And so there's always something there, but I couldn't say yes or no. It's less likely. It would be much more difficult to get DHA ramped up from ALA compared to EPA. And it doesn't take a ton of DHA to get there. And now they do have the algal DHA. So there is a vegan option of DHA. But as far as like just people's different genetic, the FADS genotypes, essentially, I think you're usually able to see significant differences in very, very large data sets because they're very, very large data sets, but the clinical difference is not there. If it could be like 0.5% different between one genotype and another. So one, you might have an average of 4% DHA and the other one 3.5%. That's not really as clinically relevant as 1 or 2% different. And so I think that's where it gets very confusing is that, yeah, we can see some of these differences in really big data sets across a lot of people, but they don't have that clinical relevance where we've seen in clinical trials. It really does take a decent amount of fish intake or a fish oil supplement to move levels quickly so that they can really be boosted up during pregnancy. Most of these studies people don't know they're pregnant until, pretty far into pregnancy sometimes. They're not recruited into studies usually until halfway through. And so if you're not recruited until halfway through, you need to have a pretty good dose to make a difference and see anything change. So some of it is just the mechanics of studying, pregnancy. But I would say I would bet on needing to get some kind of exogenous source of DHA, but there's research to be done here. And there are people who are using, because of the estrogen effect, there's actually a chance. Without estrogen, I don't know if DHA will move at all. But it's more of we just haven't seen it, I would say, in the literature.
Yeah. I think that's a kind of sensible way to approach it for the time being. I agree with you that the fact that conversion of ALA to EPA and DPA at least increases, that sort of does, I guess, emphasize the importance of these fats potentially during pregnancy. But it does seem a little counterintuitive that DHA doesn't budge much given the research findings with DHA and gestational period.
DHA budges a little bit with estrogen. It doesn't budge at all without it. That's what I would say. So I think there is a little bit of a movement for DHA, but typically it's not moved when you're doing kind of a short-term study. So that's another thing. That's a short-term studies, you have to, it's tricky. But the other interesting parts that the signals from the body that it's really wants DHA specifically is the placenta has preferentially pulls DHA and arachidonic acid from the mom's blood. From the mom's blood, it will pull DHA and arachidonic into the baby's bloodstream, so that babies will have higher levels of those two fatty acids compared to mom. If mom doesn't have it is kind of a lower source. So the baby's sucking those up. And also fat stores. So mom's fat stores of DHA over her whole life will be released a little bit to allow more DHA to get to baby, because it is a really specific fatty acid and a really specific dietary. It's just in fish, and it is so rich in the brain and the eyes that it's just like all systems go. Like wherever we can find DHA, we're going to get it there. So it's really interesting.
Hey friends, I hope you're enjoying this episode so far. A quick message from one of our sponsors who makes this show possible, and then we'll jump straight back into things. If you're familiar with my nutrition philosophy, you will know that I'm a huge believer in plant-rich diets being better for people and our planet. You'll also know that I frequently draw attention to what I describe as nutrients of focus. These are nutrients that science shows plant-based eaters, whether plant predominant or exclusive, can fall short in, which can leave you feeling run down, lacking energy, experiencing brain fog, and generally just not as vital as you'd like to be. For that reason, together with Emil, a plant-based health and wellness company, I formulated Essential 8. Essential 8 is your one-stop multi-nutrient, formulated with DHA, EPA, omega-3s from algae oil, vitamin B12, iodine, vitamin D3, iron, zinc, selenium, and calcium to perfectly complement your plant-rich diet. I personally take Essential 8 every morning with breakfast, just 2 capsules, much easier than supplementing with these 8 key nutrients individually. What's even more convenient is I have a monthly subscription, so it turns up automatically on my doorstep and I never miss a beat. To get yours, head to theproof.com/friends. That's theproof.com/friends, where you'll find a link to purchase Essential 8 that will get you an extra 5% off your first order on top of the significant subscription discount. There will also be a link to this in the show notes. Okay, back to the show. You mentioned arachidonic acid a couple of times. I want to flag that. Let's put a pin in that and come back. I think arachidonic acid might need a better PR agent because he or she does cop a bit of flack I see online from time to time.
Right.
But with regards to sort of optimal intake during pregnancy, you've kind of highlighted this couple of pieces of fatty fish a week, or 200 to 300 milligrams if we're talking about gestational period. I'm curious, so fish obviously contains both DHA and EPA. Now, when we're considering supplements, and where you spoke about 200 to 300 milligrams, I think that was just DHA you were talking about.
Just DHA.
If someone's looking to find a supplement, perhaps they don't eat fish or they're not eating it regularly, what would the kind of recommended dosage be? And would there be an even split between DHA and EPA?
There are lots of kind of recommendations from different groups, but there aren't really strong ones from kind of our medical community, I would say. It's more from the nutritional community. But the really the lowest baseline, the lowest levels are 200 DHA supplemental on top of current intake. And for me, I would rather have both EPA and DHA. So I would just want to make sure whatever my supplement was had at least 200 DHA, because now there are supplements with really different profiles of EPA versus DHA. And a lot of them are really high in EPA because there's a lot of cool stuff on that. But so that's why it's important to make sure you're looking at it and you know you're getting 200 of DHA in that supplement at least. But if it's a normal fish oil supplement, you're probably looking at a 500 milligram a day at least, maybe 600 of EPA and DHA. And that's another dietitian note. When you're looking for supplements, look on the back panel, look at the serving size, how many capsules is it? And then you add up EPA and DHA, or you just look at the DHA amount. That's individual. If it has them combined, I wouldn't get it. Don't look at total omega-3s. That doesn't matter. It does matter, but it really doesn't. What you're really looking at is EPA and DHA. And if it says 2 capsules, that's for 2 capsules. So you can't take one and think you're getting what you're getting on that bottle. So there's a lot of confusion about that.
Yeah, that's a good point, because total omega-3s, let's say, could say 1 gram, but then if you added up the EPA and the DHA, it's actually much less than 1 gram, because there's other, I guess, omega-3s that make that total amount up. I'm curious as to, and this might be going into the weeds, you can tell me if this is irrelevant, but I've seen some commentary around triglyceride DHA versus phospholipid. Is that something that people should consider when looking at a supplement? And I'm not even sure if they all kind of call that out.
Yeah. So I usually am, to me, the bigger difference is, and this is total, like EPA and DHA is ethyl ester form versus triglyceride. But the triglyceride and phospholipid DHA, I've seen in more of the brain aging Alzheimer's research and ApoE, and that because phospholipids are kind of, I don't really understand it, because I feel like it's all about absorption and all the fats get cut off from their base anyway, and then they get reincorporated into either triglyceride or phospholipid or whatever the body wants. So I don't really understand why taking a phospholipid DHA versus triglyceride would change what happens once it's been absorbed. And I haven't really seen, I haven't seen strong data, especially in the pregnancy world. I haven't seen anything. I don't even know if you can get pure DHA I guess you probably can. I don't, most of the pure DHA stuff is algal, which is triglyceride based. And that's what most of the research has been done on, but I could be missing something. In the general omega-3 space, the real difference usually is the ethyl ester forms are usually EPA and DHA. And those were the first high concentrate omega-3 supplements. They were the first pharmaceutical grade omega-3 supplements, because that was the first time they were actually able to, break the omega-3s off of the triglycerides and just put an ester group, I believe, no, alcohol group on it. So it's a much smaller package than having a triglyceride, which has a natural fish oil, only the middle, there's 3 fatty acids on a triglyceride and only one of them is an omega-3. So that's why so many of the natural fish oils have a much lower EPA and DHA content, because only a third of the fat of the omega-3, of the fat can be omega-3. But then now we have a triglyceride form where they're able to put 2 or 3 omega-3s onto a triglyceride backbone. And there seems to be can be a big difference between absorption. And that's the key, is like getting it into your body. So the ethyl ester, if you're not taking it with food, it can be very, very poorly absorbed in some people, not everybody, but enough people where it matters. And the triglyceride seems to be able to be absorbed by more people, maybe doesn't have as big of a food effect or a fat effect on absorption. But the difference between phospholipids seem to be absorbed at least as well as triglycerides. So I kind of lump those together. But as far as like the mechanisms inside the body and the different form in the pill, I just don't really understand that next step of like, if it's not an absorption issue, I don't get it. But maybe so maybe you can teach me.
Would it be safe to say that most of the available omega-3 supplements on shelf over the counter are the triglyceride or phospholipid form?
I think in Australia they are, but not in the US.
Okay.
The US, I assume so you really it's ethyl ester.
So you really need to look at the label then to kind of understand whether you need to take it with food or not, or do you just, whatever you're taking, have it with food?
That's your safest bet. This is the really fun part. There's no label requirements for the form of ethyl ester or triglyceride. If you're taking, usually the triglyceride forms in the US will be advertised as a triglyceride form. It's like a front of package. It's not anything official. It's just like this. It's marketing. And that's useful. If it doesn't say that and it's a high concentrate, I assume it's an ethyl ester. Most ethyl ester products do not say it's an ethyl ester, as far as I know. If it's a pharmaceutical grade, I think all of those are ethyl ester. Those are like 4 grams a day. So you end up getting enough in there. But yeah, it's not clear.
Sure. So as a kind of safety step, if you're having a supplement, it seems like a wise idea to do it with a meal. You mentioned algal oil there, and of course that's starting to pop up and becoming more available and more popular. Is it just as effective as fish oil, DHA and EPA in terms of changing omega-3 index?
Yeah, it's really effective actually. And DHA is more powerful than EPA in changing the omega-3 index as well, just in general. But it's really potent and absorbable.
Yeah, I wonder, I know this is not your area of primary research, but given how important omega-3s are and all of your research into them, particularly during pregnancy, and thinking about making these more available within the food system to get omega-3 index up across an entire population, do you ever give any thought, I guess, to how that can be done in a kind of sustainable manner with 8 billion people, and whether it's sustainable enough to do that through fish and everyone eating 2 pieces of fish a week, or whether we need, other options, be it algal or additional options to kind of work DHA and EPA into the food system?
Yeah, that's a really great question. And it's one of the reasons why I'm kind of glad to see this, the effect on preterm birth happening at an earlier stage than an 8% omega-3 index, because it feels more doable on a public health scale. It seems like basically all the food we eat is at an unsustainable pace, and fish is no different. But yeah, it's a major concern like when you do the math, you're like every pregnant woman eating 2 servings of fish at least a week. And what does that do to our fisheries? But I would say that there are a lot of smaller fish, which tend to be a little bit easier on the system. And also, usually it's like the sardines and the herring and the stuff we aren't used to eating. At least I'm not. But if I could get myself to like it, you can buy tins of them and they're easier to just get on your plate. Salmon, of course, is the big one. And it's being farmed quite a bit, which has its own issues and pros and cons. I can't be I'm fairly agnostic about, like, there are so many different things people are considering with their food choices and their fish choices. And I don't want to add extra stress on that if it's a piece of farmed salmon, that can have a very high amount of EPA and DHA, it can be more than wild. But just because they give them so much fat in general. So it's maybe not like as good of a profile, but I kind of just want to give people space to say, however, it works for you and your values, and especially pregnant women, understanding the fish issue with pregnant women, where there's a lot of fear around it because of the mercury issue. And many of the fish that we eat on a day-to-day basis have low levels of mercury, where you would be able to eat fish daily and it would be fine. They've done studies where they've shown that children from women who've eaten fish in island countries, like the Seychelles, those children have excellent cognitive outcomes despite having higher omega-3 and higher mercury levels, because often there is going to be mercury in the fish, but it's just not at a very high level. And so there's a lot of fear around fish in general. And so if that's happening, I'm like, just take a supplement. Don't force yourself to eat fish when you're pregnant, because you can hardly make yourself eat anything you don't want when you're pregnant. And the supplements are typically quite clean and they've been processed. They don't have any contaminants. it's kind of just confusing on what you need to get and how much you need. And sometimes it's hard to take supplements when you're pregnant. Sometimes it's just hard to get anything down. That's kind of a separate piece for pregnancy. That's just, it just adds to the stress of making a food decision, which I feel we just in pregnancy need to try and de-stress as much as possible. But if they're like, when it comes to like the sustainability of the food system, I think fish is a great option. It gives you a lot more than just omega-3. And if you can eat those smaller versions or choose to go with smaller fisheries or places that can track the fish through the whole process, that's great. That at least gives you a little bit of confidence that you are supporting a local fishery or a smaller fishery or something that you think they're doing it, quote, the right way. But at the same time, all the fisheries are interested in having a fish source in the future. So it's really like, it's a tricky decision for everybody, because with everything around basically like climate change and our individual decisions, there's always this, you can get really stuck in what should I choose? And there's already like, that doesn't even bring in cost and taste preference and how to cook it and how to store it. And so for me, it's like, get some seafood on your plate. I think it's a great variety. Whether or not, the sustainability question is definitely outside of the realm of my expertise. I'm also concerned about it, but I also think that the fish decision it's one of the pieces of a lot of different decisions you're making about one food. And so I kind of go to my RD head of like, that's a lot of pressure on one food decision. And so I think if you're open to having fish, I think that's really great. If you're not, supplements are also a great option. They both do a good job of raising omega-3 blood levels.
Yeah, I think that's a fair position. And it's almost a different question for an individual versus a society, a population. And being able to say, hey, here's what the science shows. There's clear benefits for DHA. Now, here's the sustainability piece. What do we do on an individual level that feels right for us? There's a few different options there, fish oil or an algal oil supplement. Take your pick, what feels right for you based on your circumstances. What do you have access to? What can you afford is within your budget? And then on a population level, getting serious about DHA and the science and realizing that index levels are low. What are we going to do from a public health perspective, and how will we do it responsibly without exploiting the environment and fish, for example?
Yeah.
So I think we're kind of aligned there, and I'm glad that there's a few options for people to kind of choose and land with what feels right for them. I'm curious as to supplements. If someone was to choose a supplement, I think sometimes there's this idea with a lot of things in nutrition, when something's good, more equals better. And I wonder, are there any side effects or risks associated with, say, consuming too much of these long-chain fatty acids? I've heard various people talking about different things, one of which that I see quite a bit is increased risk of bleeding. And I think I've seen people talk about that potentially in the third trimester. Is there anything here that you think is kind of valuable for people to be aware of?
Yeah, definitely. So I think what's interesting is like every nutrient has a deficiency state and a toxicity state. And so the same is true for omega-3s. There's a point where it's just too much. And the quote toxicity symptoms, even though they've never been truly defined and a target point's never been truly defined for that. It really is kind of the excessive amount, the excess of the mechanism of action that you want to see. So better blood flow, reduced platelet clotting, improved circulation essentially is really good for most people. And then if you go too far, clotting time can decrease for sure, but typically clotting time will decrease, but not completely be shut down. It's not like warfarin. Warfarin is much more dangerous than fish oil as far as like a bleeding risk. And we've actually, my dad has actually compiled a paper on bleeding risks in lots of different areas and operations, and in other outcomes, and pregnancy is one of them. And from what I've seen, risk for severe bleeding events in pregnancy is not increased with high dose DHA, whatever we've tested it up to, at least 6 grams of fish oil a day, not necessarily EPA and DHA. It doesn't mean, and in one of the studies I'm remembering, the fish oil group had technically like a greater milliliters of blood lost at delivery than the other groups, but it wasn't a significant bleeding event. It was just a little bit more blood was lost. And so there's a difference there. And then across the board, we've not seen that significant bleeding events are related to high dose fish oil. But it is a slower clotting time. So you might notice in yourself that you bleed more because you are bleeding more, but you do end up clotting eventually. And so that's just that's part of the deal when you get, it's one of the good things about omega-3s. So the other things with really, really high fish oil, it's mostly anecdotal. At this point, we have an upper limit set on ours as 12% omega-3 index for, you really don't need to go above 12%. And that's actually sort of arbitrary. We just don't have a lot of data above 12%. But if you look at fish eating cultures like Japan, which is kind of our classic culture that we look at, most of those people, based on their current diet, where they're eating fish every day or most days, they can start out at a 8 or 9% naturally. And then they'll often add omega-3s on top of it, or they've done a study, a jealous study, where they added omega-3s on top of that. And they were getting people up to 11, 12, 13, 14%. And they continued to see improvements in heart disease outcomes. Another kind of like side story on this that I just find so interesting is my brother used to be stationed in Japan as a doctor with the Air Force. And he was in a small town, and he found that the local hospitals, the Japanese hospitals, when someone came in with like a heart attack symptom, they would not immediately give warfarin or immediately give a blood thinner, because they didn't need to, because that was not the issue for most people there. They didn't have that clotting issue because they all had a lot of other things happening. But one of the things could have been, as a population, they're at a much higher, they're already basically all at that 8%. So that anecdote always just sticks with me. But so that bleeding, it all comes back to that bleeding piece. And so, of course, you can, with the high dose supplements these days, we could start seeing people really get up there. And at OmegaQuant, we do see people who are 15, 16, 17% sometimes. Even the highest I think we've ever seen is 22%, which is higher than a dolphin. But as far as we know, those individuals aren't having major issues, but we aren't necessarily tracking them. So we don't, that's just a lack of data. Is what's that point where it's like, you've gone too far? But I think you could get to a point where it's just like, the bleeding is really uncomfortable. But for most of the studies that have been done, those severe bleeding events are not increased on the fish oil arm.
Mm-hmm. Do you have any kind of sense as to what dosage of DHA EPA would be required for the average person in, say, America or Australia to get to an omega-3 index of, say, 12%? And I guess second part of that question, and of course, only if you mind sharing, but I'm curious if you supplement and what kind of dosage you've chosen for yourself.
Yeah. So we did a paper with Rachel Walker from Penn State. And we did ask that question of like, how much should we tell people to dose? And so we were able to take 14 studies where we had done the blood analysis and they had just given a standard supplement for whatever outcome. And so we're able to get raw data from 14 different studies. And we just used a biostatistician and had him make a calculator. And so we could say at this, if we start at a 4%, how much do we need on average to get to 8%? And the 4 to 8% number that he found was actually different between a triglyceride and an ethyl ester form. Those two were giving such different numbers that it's two different equations. So the triglyceride number is about 1,450 milligrams of EPA and DHA a day, which does kind of go with about a serving of oily fish a day. And a salmon serving can be about 1 to 1.5 grams of EPA and DHA. And then for ethyl ester, it's 2.2 grams a day. And so it's quite a bit higher for the ethyl ester. And we think part of that higher dose is the absorbability across the board. But then in certain people, it is very dramatic how much they can absorb ethyl esters. But it's not everybody. So those are the two numbers to go from 4 to 8%. On average, the US is about 5.5%. So it's more closer to 1,200 or a gram probably for most people. If you want to, that's to get to 8%. To get to 12%, our data ran out at about 3 grams. We didn't have any studies that went above 3 grams in our calculator paper. So we don't have recommendations above, I think it's 3.2 grams. But it should be 3 grams should be, that would put people above 8 for sure. Mm-hmm. We don't usually we see 12% as kind of an upper limit where, again, it's like, we don't know if there's much. It seems like you get most of the bang for your buck going from low to 8 to 12%. Mm-hmm.
Sure.
And going from 8 to 12 or 15. I don't know if the the cost benefit ratio is there. Mm-hmm. And it's like, more isn't always better. How much does it cost for you to supplement to get up to that point? I would also say, so for some, and I'm kind of speaking about the general public, there are certain medical conditions that some people are using high dose fish oil to kind of treat themselves or treat symptoms. And that's a different question than just generally getting to 8%.
Yeah, we spoke about the Reduce It trial with your dad, and I think that was a 4 gram serve. Mm-hmm. You mentioned there 1.2 grams for the average person, speaking, very broadly, to get up to about 8%. Just to be clear, that 1.2 grams, is that active DHA and EPA, or is that the total omega in that dose?
Specifically EPA and DHA.
Got you.
So not total omega-3s. We had to take, for our calculator, we took EPA and DHA from those supplements and put it into our database. So we always operate off of EPA and DHA because there's so much variability. Like, that's the only way we can reduce variability, is to just focus on those two. And we are aware that there are other omega-3s, specifically DPA, which are doing things, are important. But adding them into, on a practical level, EPA and DHA are the signals from fish and fish oil that are really potent and strong, and doesn't add a whole lot as a signal or as a marker to include DPA in that, even though biologically it's very likely acting as well. Okay.
And that brings us back to the point we made earlier about the importance of looking at DHA and EPA levels on the label rather than total omega-3s. Mm-hmm. One thing that we haven't asked you yet related to pregnancy is the ideal time to get your omega-3 index up to, say, 5%. Are we talking about a sort of protocol that a woman would start after she discovers that she's pregnant, or is it kind of safe to assume that anyone who is of a childbearing age, even if not pregnant, should aim for kind of 5% as a baseline?
Mm-hmm. Yeah, I would say the second. I would say it's like folate a little bit, and in other ways it's not. But ideally, everyone would go into conception at least a 5%. I like thinking of it as kind of a baseline level for being replete. It's not necessarily our optimal level. So going in, because that's where that Carlson paper, they found that women coming in above 4.8% had a lower risk for preterm birth across the board than even the women getting 1,000 milligrams, but they came in lower. So coming in, it's not that hard to get to 5%. You don't have to take 1,000 milligrams to get to 5%, but getting up there, getting your stores up seems to be really important. But I will say most of the studies have been done where, like I was saying before, they're not recruited into the study and starting supplementation till the second trimester. Just because it's pregnant, studying pregnant people is difficult. And so in those studies, they found that providing omega-3s at that point does something. It matters. It changes outcomes. It changes the gestational length. And the other piece of the biological piece is the third trimester is when the majority of the fat is being transferred to the baby. So that includes DHA. So that having DHA on board just in the second half of pregnancy is absolutely better than nothing. And that's where most of the studies have shown efficacy. But those are going to typically be, again, like a higher dose at that point. At that point, you're probably going to want 1,000 if you haven't really been doing anything at all before that. And again, that's where if we could test at that first trimester or early in the second trimester and see where you're at. And if you're at 5%, then you keep doing what you're doing. You shouldn't need to add on and add on and add on. I don't think it would hurt. I don't think it's necessary. But if you're low, that's where you really need to get that dose up. And we would have, this is the way that, that life works, where it feels like the rich get richer and the poor get poorer, where if we could just focus on the people who had low levels, that would do so much more for reducing preterm birth risk than people who are coming in with okay levels, but then they bump them way up to like 8%. They're probably not really at major risk for preterm birth because of DHA deficiency in the first place. So it's really that group that's coming in low that has no idea where the benefit is it's in that group.
So would it kind of be safe to say, in an ideal world, you'd be able to quantify this with an omega-3 index for everyone and understand where your baseline is at. But would it be fair to say if your baseline diet going into pregnancy hasn't included a lot of fish, hasn't included a fish oil supplement or an algal oil supplement, you might be coming from quite a low baseline. And in that circumstance, you may need more than 200 to 300 milligrams of DHA in order to get you to a sort of 5% index level?
Yes. So we did a study Cornell University ran a study where they were looking at choline, actually, and that was their intervention. But they gave everybody 200 milligrams just standard and controlled the rest of the diet in pregnant women in the third trimester. And we were able to analyze some of their blood levels. And most of those pregnant women came in, I think, in the 4% range. And it was a small study, but about 20 people. But they were about 4%. And with 200 milligrams, the average went up to 5%. That was actually in the lactating group. Actually, I think they came in at 5% and were up to 6% in the pregnancy group. But if you don't eat fish, what we were seeing is that in non-pregnant women, those women came in at about 4% and they ended up above the 5% mark. So a non-pregnant person, if they want to increase their omega-3 levels over the course of 4 or 6 months, you can do 200 or 300 milligrams a day, which is kind of a typical prenatal pill. Or you can start adding fish into your diet with some regularity, and they need to be higher omega-3 fish. But you need to have a good amount of, like, half a year probably on a lower dose to really build up to that. If you don't have a half a year, then you need to have a higher dose. You want to go for 1,000 plus fish, or plus like if you want 2 or 3 servings of fish, but take a higher dose supplement, between 500 or 1,000 or more if you want to, of EPA plus DHA is fine for those numbers. But getting that before you're pregnant, you can really ramp it up a little bit faster if you are giving a higher dose. But that low dose over kind of a longer period of time works too.
Yeah, that's a really important point. I guess if you just found out you're pregnant, you don't really have 6 months to slowly get your levels up to optimal. And that's a point your dad actually made, and I think that's a valuable one, is if you have tested your omega-3 index and then you go and make some changes, be it supplements or food, it can take this 3-4 month period in order to allow sufficient time to see the effects that has on the red blood cell membrane content.
Yes. Yeah. Red blood cells turn over about every 4 months. And so that's kind of the time you want to give that dietary change, to see that's, there's still some increase typically after 4 months. But that you kind of see your steady states. Then, if I'm taking 1,000 milligrams a day, that lands me at 7% omega-3 index, maybe. If you, and you're probably not going to go up from there. So then you can know, okay, if I actually want to get to 8, I do need to up my dose a little bit. That's where the steady state's important, because it gives you the information of like, I'm probably not going to go up anymore from here on this dose. And the dose will really fairly closely match. Once you figure out what you need, dose is really important, but there are other things that are affecting the omega-3 index that are somewhat unknown, but also we start to see this actually kind of in the athlete population, where omega-3s can be depleted over the course of a season, with kind of that intense inflammatory burden of being in season. It seems like it can be used up. So there's that aspect that could increase your needs a little bit, or if you're like supplementing at this level and it gets you here, if you have some kind of event happen, like pregnancy, we do see in pregnancy, the women's levels can go up and up and up, and postpartum, they drop like a rock. And that's partially because of breast milk. It's going into their breast milk. So there are some patterns also that can interrupt that relationship between your supplementation dose and your level. But if you're kind of normal status, then that should 4 months should show you what you need to know.
Yeah, that kind of emphasizes, I guess, keeping a track of what you're personally doing, and then being able to objectively quantify where your levels are at over time, and you'll hopefully be able to determine the right, or specific right, I'll put in quotation marks, sort of protocol for your biology. I'd like to slide over here as we come to the end of this to the postnatal, neonatal sort of infant period. I know that you've done some research, and you mentioned before looking at the DHA content of breast milk. I'm interested in how important that is, how important the DHA content is in infant formula when it comes to sort of the first 4 to 6 months, where the baby is reliant on either of these as a sole source of nutrition.
Yeah, good question. And I would say formula is not my expertise. I was really focused on breast milk literature. But of what I know, and what I saw, the really striking studies that showed DHA and arachidonic acid are actually both really needed in formula. The more obvious outcomes come from the premature babies. And part of it is they are more fragile, and their outcomes they're able to look at more dramatic outcomes. The closer to term a baby is, the more robust it is. And I think the harder it is to show true differences between different types of formulas. But I know that in Europe, they made they basically required DHA to be a part of infant formula. I'm pretty sure that didn't happen here in the US. But there was kind of an uproar because they didn't require arachidonic acid as well. And there were some studies showing that high DHA without that accompanying arachidonic acid level in the formula was actually a worse outcome than a low DHA. And both of those fatty acids are incredibly important in brain building. And so I would say generally, I don't know. I mean, 0.32% DHA used to be kind of the level that was in formulas. I think it's higher now. In breast milk, that's the worldwide average, or it was, 10 years ago, probably, when they did kind of an assessment. And we found that 0.32% in breast milk was kind of that low cutoff for being replete as well. DHA in breast milk. But the arachidonic levels in breast milk, those usually stay pretty steady at 0.6%. So they're usually higher than DHA anyway, but DHA can range from less than 0.1% to 1%. There's huge variability. But in formula, I think making sure that having both DHA and arachidonic together, that's the big takeaway that I have from that. I don't necessarily have as big of a message about the dose in the formula. And also that the effects are maybe more apparent in premature babies versus term babies.
Sure. So in order to achieve that kind of, I guess, minimum level of 0.32% of DHA in breast milk, what's the kind of protocol? Is the advice that we went over before in terms of consumption of fatty fish and or supplementation at those levels, is that sufficient to ensure this much DHA in breast milk?
Yes. So this Cornell study I was talking about, they also did a lactation arm. So we actually were able to do the same thing where we just were able to look at breast milk and red blood cell levels in lactating women, taking 200 milligrams of DHA a day. 200 was not enough to increase their red blood cell levels. It maintained their levels, but it increased their breast milk levels. So all of it was going into breast milk. And I would say you probably have higher needs of DHA during lactation, where it's at least 300. That's at the very least. And part of that is growing a baby is a lot of energy, but creating milk is a huge drain. And as baby gets bigger and bigger, the needs get bigger and bigger. And the DHA levels in breast milk are even more tightly tied to current intake. The stores aren't as being used as much. It goes up and down a lot faster, like more like plasma than like a red blood cell. And so I would say in lactation, I would do it. I would have it. If it's at least 200 in pregnancy, it's at least 300 in lactation. But the needs appear to be higher in lactation to also to replenish mom, because a lot of times mom is depleted and needs to be restored. And so there's a higher level needed for mom's body, and there's a higher level needed for like making breast milk with DHA in it. And as we know, like the baby's brain is growing a lot during this time point, and their whole everything in their body is still needing a lot of nutrition. And DHA is providing a really important nutrient for baby in that time. So yeah, I would say DHA needs are higher in lactation than pregnancy.
Mm-hmm. And I know that you said you're not an expert on infant formula, but I do often, and you have mentioned arachidonic acid a couple of times, and when I hear infant formula come up, I often see parents expressing concern about omega-6s. And linoleic acid usually comes up, which is, I think soybean oil is often one of the ingredients. And so I'm kind of curious to hear from you. Do mothers need to be concerned at all by omega-6s in infant formula, given that breast milk contains omega-6s? Do they need to pay attention to the label? Do they need to look at the omega-6 to 3 ratio? Is any of that important?
I would say, so linoleic acid and arachidonic acid are the main omega-6s, but they're quite different. And in the body, your linoleic, having higher linoleic acid levels does not mean you have higher arachidonic acid levels. Arachidonic acid seems to have a much more metabolically controlled level. So the body is making sure it has exactly as much arachidonic acid as it needs. And so increasing linoleic in the body or in the diet typically does not increase arachidonic. One of the only things you can increase arachidonic acid by supplementing it. I don't know why you would do that. It's in meats and eggs primarily as a dietary source, but that isn't even as powerful as the metabolic control in the body. So having a formula with linoleic acid does not mean it has arachidonic acid, and they are two different fatty acids with two different functions. And so I would say the arachidonic acid, something you want. Linoleic acid it's usually, that was the filler fat from what I understand. Like that was just, we needed a fat source that was able to work within the formula. And that's typically what they use. But I really don't have a lot of the data on this. If the omega-3, omega-6, or if the omega-6, omega-3 ratio is low, it's typically because there's not enough omega-3 in it. Mm-hmm. And that's usually the area of concern. But a lot of people, the omega-6, omega-3 debates are raging in the nutrition community, and there's still not really clear agreement in the scientific community. And then it's also just in the public. It's a very confusing topic. Mm-hmm. And part of the problem is they kind of work with each other. So when you increase your omega-3s in your body, when you increase your omega-3 index, typically your arachidonic acid goes down a little bit and your other long chain omega-6s go down. So if when you change one, the other one changes a little bit too, it's hard to know which one is important to change because two things are changing at once. And so that's one of linoleic is not one that's affected by increasing your omega-3 index because it's 18 carbons. It's shorter chain, typically. But yeah, I don't really want to be too much into fear about it because I don't know. I don't know enough about it, but I know like basic safety has been demonstrated with any formula on the market, I would say. But as far as these fatty acid levels, it's way beyond my depth at this point. But I'm just focused on the DHA and arachidonic being present.
Sure. Okay. So that's instructive to look at the label, make sure that both DHA and arachidonic acid are there and have been included in that formulation. Mm-hmm. I want to circle back to a point that you made earlier. I think you were mentioning an Australian randomized controlled trial, if I remember correctly, that was looking at postnatal depression, but also some outcomes looking at cognitive function in young children. And
yeah,
I'm interested.
I think they followed up their study. Yeah.
Yeah, you kind of said or alluded to the fact there was some conflicting findings there with regards to ensuring adequate DHA intake through pregnancy and while breastfeeding. Do we know how this actually plays out in terms of the child's cognition as they get a bit older and their IQ?
Yeah, I'd say it's a really mixed bag research-wise at this point. And I think there's a lot of reasons for that. One of the reasons to me is that it's so hard to study childhood cognition. Like the things that researchers do to try to assess this, it's just such a difficult marker to analyze. So that to me introduces a ton of variability in this. But there's definitely been I think it was the Domino study, and they followed up over at least 7 years. I think I remember it was one of theirs. And they wouldn't see any differences between the groups. Sometimes they'd see a difference on like one scale, and then they wouldn't see anything in the next year. it's really kind of back and forth. It depends on how many people they actually can follow up with, because that's a big deal. Like who are the people that continue to follow up with a research study? That's not the same population as your original study. It's a subset. And so that's going to affect your outcomes. It also is tricky because it's like you're looking at what happened in pregnancy and then following out out to like 2 years and 5 years and 7 years. And it's like, there's also what happens in between for those 7 years, what they've been eating. So, if they were getting omega-3 levels on these kids, then you could really look at, the behavior scores and the omega-3 levels, on a continuous basis, which is usually where we do find effects. It's hard to find effects when you're just analyzing data based on assigned groups, because we know that there can be major issues with compliance in studies. There also, and that can be revealed if you test their blood levels, because blood levels are they're pretty regular. If you're giving people a good dose, it goes up.
Yeah.
It doesn't lie.
Yeah, they don't tell fibs, I should say.
Yes. And I think people are trying to be good participants. They don't want to do a bad job, but that's why you test. And then you can take your blood results and you can look at outcomes based on what blood levels were achieved. And so this actually happened in that St. McCrady's group. They also just ran the biggest study done on pregnant women. And they published in 2019, I think. And they gave 800 milligrams of DHA to one group and 0 to the other group. And they found that they didn't see a difference in preterm outcomes. It was over 5,000 women. It's so much bigger than any other study that was done. So it was quite a feat. But when you look at the blood levels that were achieved in the group that took 800 milligrams, it was around 5%, when it should have been probably 7% at least. And then the other group was around 4%, 4.5%. There was not a huge blood level difference on average between the two groups. So something happened. There's potentially a compliance issue, and that would explain a non-effect. So they did a secondary analysis based on baseline levels, and they found the same thing, where women who were very low had an effect. They were protected by having a higher dose of DHA, but if they were higher baseline levels, it didn't matter what group they were in. So, but that's part of that whole issue of compliance and research studies and outcomes. And if you're looking at outcomes years down the road, you really do need to be measuring blood levels to have any idea of what's going on omega-3 wise, in my opinion.
Yeah, and it highlights a great point in terms of making some of the observational research that's done a little bit more robust. What essentially that domino study, what you're talking about there, I'm not 100% familiar with it, but if I'm hearing correctly, it was a randomized controlled trial. Then people started free living. There's a whole lot of other variables. It's almost an observational study at that time. And how can you actually more objectively understand the tissue levels of omega-3s in these children that are undergoing these cognitive tests? I think that's a really good point. And I actually hope that these tissue biomarkers, and we're already seeing it, are used more and more and more in observational research to kind of support the food frequency questionnaires and just make the whole study a little bit more robust.
Absolutely. Especially because it's well established that you can do the fatty acid blood markers from a dried blood spot. So the collection is not an issue. You can send it to people. And so it makes it accessible for any kind of research.
To wrap this up, I think a sort of high-level summary might be useful for folks listening who are thinking, gosh, there was a lot of dosages in milligrams and DHA, EPA thrown around in this conversation. I'm of childbearing age, or I'm pregnant. I just want to hear a kind of very simple list or protocol what I can walk away with and implement in my life?
Great question. I think boringly following a lot of the recommendations is a great idea. 2 servings of fish a week before you get pregnant. I mean, that's really good for basically everybody. If you're not a fish eater, I would recommend at least 200 or 300 milligrams of DHA specifically. If you want to have fish oil, you're probably going to look at about 500 milligrams at least. And doing that, I mean, like with anything, doing things regularly, the consistency is everything. And so if you're pre-pregnancy, you can do that. If you're in pregnancy and you think, I definitely have not been eating fish or taking fish oil, I would look to get a higher dose of fish oil. I would maybe go for 1,000 milligrams of EPA and DHA, or 1,000 milligrams of DHA, which usually is like a 2 or 3 gram dose of fish oil. So it really depends where you are at in the process. But if you're coming into pregnancy and really want to bump your levels up in pregnancy and you're halfway through or something, you probably do need a higher dose. There shouldn't be any safety concerns at the 1,000 milligram DHA level. And that was shown in these recent studies. And so finding that product is not going to hurt, and it's going to be beneficial for a lot of different aspects of the pregnancy. And so that's what I would go with.
Okay. And final question, if we were to kind of zoom out a little here, and perhaps I'll ask you to put your registered dietitian hat on as opposed to the omega-3 researcher. We know that during pregnancy, there's a number of important nutrients and food groups, and I'm sure people could create a case for saying that pregnant women are not consuming enough fruits and vegetables or fiber, etc.
Absolutely.
I'm kind of just interested in overall, in addition to omega-3s, what are some of the other low-hanging fruit, the parts of just overall nutrition that you think pregnant women should focus on a little bit to ensure they are achieving nutritional adequacy or an optimal kind of nutrient status going through that period?
This is a really great and tricky question because this is a double-edged sword. I think a well-balanced diet of lots of fruits, vegetables, kind of whole foods, whole grains. I think some amount of meats provides some really important nutrients like omega-3s and fish, and meat. Omega-3, vitamin D, and B12 are all and choline are all present in fish. Choline's those 4 are actually really important in pregnancy. D, B12, and choline are found in other things besides fish, and omega-3s are kind of specific. But I would say I'm not as broad as I could be, but I have learned more about the vitamin D and choline importance during pregnancy. And choline and omega-3 actually have a really kind of integrated metabolism, especially during pregnancy. But generally, it's a well-balanced diet should provide those things. Having, unfortunately, prenatal supplements don't necessarily include choline or omega-3s. And so that's why those two kind of jump out at me, as they aren't kind of they're not the folic acid that is present in a lot of the already pretty ubiquitous as an option. And so, yeah, the double-edged sword piece is you can drive yourself crazy thinking about like, oh, if I don't get the optimal nutrient, I'm going to damage my baby forever. Not true. Our bodies are very resilient. They are going to do everything they can to make baby as well as they can. And it's not a perfect world, so it won't always be perfect. But I have a real it's really hard for me to talk about this without thinking about that other side of it, is if you didn't think you got enough of these nutrients, blaming yourself for something that happened in your pregnancy, when these are just like, they're part of the story. They're part of the picture. They're not the whole picture. Try and do your best, but also it's bigger than this. It's more complicated than this. And it's not like you won't be permanently damaging your child if you don't get enough choline necessarily. But there are things we've seen biologically that really are helpful. And like for DHA, we see your body will release as much DHA as possible to get to baby. So there's redundancy for a lot of this stuff. But that, on the same note, does not mean that getting enough of these things through our diet, through extra supplemental nutrition, doesn't help us feel better in the process, doesn't help the process go a little bit easier. So I can never give a straight answer, but that I just know that I felt that when I was pregnant, is that overwhelming stress about all the different nutrients that I may or may not be getting enough of. It was just, and sometimes that stress is exactly what we don't need. So calm down. You're doing great. Try and eat a lot of different kinds of good whole foods, and talk to your doctor about it.
I love it. Well, thank you very much, Dr. Harris. This has been super informative, and I really appreciate that piece of perspective there at the end. I think that's an important reminder, and you really did put it beautifully. So thank you for that. If folks listening would like to get in touch with you, what's the best way for them to do so?
I'm through OmegaQuant. Most of my presence is through our website and through, we do a little webinar with my dad and I called Omega Matters. I'm available to be contacted through our website, I believe. Just doing info at OmegaQuant, and it'll get shuttled to me. And otherwise, I think I'm on LinkedIn. I'm not really active professionally on social media. Otherwise, I try to keep my sanity. But yeah, you can contact me through either LinkedIn or the OmegaQuant website. Okay,
great. I'll put the links to those in the show notes. And yes, I agree. Social media can sometimes not be that great. I appreciate you taking.
Yes. There's a lot of good stuff. I have learned a ton through people who are keeping up with research on social media. So I appreciate it. Yeah. I'm just not doing it.
It's got its good and bad parts. For sure. Okay. Well, I appreciate you taking the time today to educate us, and I hope that we can continue this conversation down the line.
Yeah, that'd be great. Thanks. This was fun.
Thank you for joining me for this episode and your interest in science-based conversation. I hope you enjoyed it and found the information covered interesting and instructive. If you did and you'd like to show your support for the show, please subscribe to our YouTube channel, where you can stay up to date with new episodes and watch them in video format. Yes, the full-length videos. Please also consider subscribing to the show on the Spotify and/or Apple Podcast app, wherever you enjoy listening to podcasts. You can also leave a review on Apple or Spotify. Again, a great way to support the show and make our content more discoverable for others to enjoy and learn from. If you have any comments about the episodes, suggestions for future episodes, including guests you'd like to see on the show, or questions that you'd like to have answered, please leave those in the comments section on YouTube. I myself and my team will take note of these comments when planning future episodes. Finally, the best way to support the show and receive discounts on products we love is by checking out our sponsors at theproof.com/friends. Enjoy your week, stay well, and I look forward to catching you in the next episode.